Welcome to another edition of The Explainer. I am so incredibly thrilled you're here today because, honestly, we're looking at something that fundamentally shapes human survival. Right? Have you ever wondered how scientists are able to develop these absolutely resilient super crops, plants that are disease-resistant, drought-tolerant, and high-yielding, and do it so remarkably fast? It literally seems like magic, but it's actually a brilliant application of molecular biology. Today, we're demystifying a completely groundbreaking scientific process. It's called marker-assisted selection, or MAS for short. So, to really understand the sheer genius of MAS, we've got to ask ourselves this core question. How exactly do scientists build better crops faster? Just think for a second about the enormous, totally unprecedented pressure our global agricultural systems are under right now. We desperately need crops that can survive harsh droughts and fight off devastating diseases. But historically, developing a new plant variety took years. Sometimes decades of just waiting around for results. We simply don't have that kind of time anymore. We need rapid, highly accurate solutions. Okay, let's dive into this. Here's our roadmap for today. Number 1, the core concept. 2, molecular markers. 3, how MAS works. 4, advantages and limitations. And finally, 5, real-world success stories. All right, starting with section 1, the core concept. Decoding the DNA of plant breeding. What we're really talking about here is a massive paradigm shift in agriculture. It's huge. We're essentially moving away from thousands of years of traditional farming techniques and stepping directly into the realm of modern genetics, literally decoding the very DNA of the plants we rely on to survive. For centuries, traditional breeding was basically a giant game of wait and see. Breeders would cross plants, plant the seeds, and then, they just had to wait months or even years for the plant to fully grow up just to see what it looked like on the outside, to determine if it actually had the trait they wanted. Marker-assisted selection totally changes the game to predict and select. Instead of waiting for the plant to mature, breeders can analyze the plant's DNA when it's just a tiny seedling. They select based on the internal DNA, which makes the whole process exponentially faster, way more accurate, and just far more efficient. Moving on to section 2, molecular markers, the genetic signposts. So if breeders are picking out these desired traits at the seedling stage using DNA, what are the actual biological tools that make this early detection possible? How do they know what they're looking for in a tiny little green sprout? Well, the answer lies in molecular markers. These are specific DNA sequences located very close to a gene responsible for a specific trait. And this brilliantly illustrates the core mechanic of the science. The markers themselves don't actually control the trait. You can think of them exactly like genetic signposts on a highway. They simply point scientists directly to the desired gene they want to keep. If the marker is there, the breeder knows for sure the gene is almost certainly parked right next to it. Now, scientists have developed a bunch of these signposts over the years. You might see older acronyms in historical research, stuff like RFLP, RAPD, AFLP, or SSR, but hey, don't worry about memorizing those. The ones I really want you to focus on are SNP and KASP markers. These two have absolutely emerged as the modern standards. They're the primary go-to tools widely used in today's modern crop improvement programs, mostly because of their high efficiency, their speed, and their incredible reliability. All right, let's hit section 3, how MAS works, the step-by-step blueprint. Now that we know what the tools are, let's actually look at how they're used in practice. We can break down the laboratory and field workflow to see how a new crop goes from a mere idea to a reality in 4 distinct steps. Step 1, breeders start by crossing 2 parent plants. One parent has the desired gene, say disease resistance, and the other has superior agronomic performance, like a naturally high yield. Step 2, from that hybrid population of offspring, scientists extract the DNA. Step 3, they perform marker analysis on that extracted DNA, specifically hunting for those genetic signposts we just talked about. And finally, step 4, they immediately identify and select only the plants carrying the target gene to advance to the next generation. Because all of this happens at the seedling stage, it saves an immense amount of time, land, and resources. You're literally only growing the plants you already know are winners. Which brings us to section 4. Advantages and limitations, balancing the science. Now, of course, no scientific breakthrough is without its hurdles, right? So we need to impartially explore both the incredible benefits and the realistic limitations of marker-assisted selection. Let's kick things off with the advantages, which I have to say are substantial. As we've covered, it results in much faster breeding, incredibly high selection accuracy, and allows for that absolutely crucial seedling stage selection.
But what's really interesting about this approach are two specific breakthroughs that basically change everything. First, MAS is not affected by environmental conditions. In traditional breeding, a random flood or a weird heat wave could completely mask a plant's true genetic potential and ruin the whole experiment. DNA analysis totally bypasses that risk. Second, it allows breeders to pyramid multiple resistance genes into one single variety. You can essentially stack different genetic armors into one super plant. How cool is that?
However, we absolutely must look at the limitations too. This isn't exactly a weekend project you can do in a standard greenhouse. MAS requires expensive, highly specialized laboratory facilities and incredibly skilled manpower. Furthermore, it completely relies on reliable molecular markers already being available for the specific trait you want. And perhaps most importantly, this is a big one, MAS is highly effective for traits controlled by one or just a few genes. But when it comes to highly complex traits controlled by many interacting genes, like overall grain yield, MAS is actually less effective. For those complex traits, breeders actually have to zoom out and analyze the entire genome at once using a totally different method called genomic selection.
Moving into Section 5, Real-World Success Stories, MYS in Action.
Let's step out of the laboratory for a second and into the actual fields to see how this builds up and protects crops growing around the world today. Because this is where the science literally saves lives by protecting our essential staples. For rice, breeders used MAS to introduce the sub-1 gene, which gives the plant submergence tolerance, allowing it to survive devastating floods. In wheat, they've successfully integrated the LR and SR genes to provide vital resistance against rust disease. For maize, it's been used to create resistance against the maize streak virus. And even in everyday produce, like the tomato, MAS has successfully provided resistance to the tomato yellow leaf curl virus. These aren't just theoretical concepts in a textbook. These are real, successful, highly official crop varieties feeding people right now. When you step back and look at the whole picture, the stakes become very, very clear. By combining genetics with molecular biology, marker-assisted selection has absolutely revolutionized modern plant breeding. It is our vital, highly precise tool against the accelerating threats of climate change, rapidly emerging diseases, and the ever-present daunting challenges of global food security. It ensures our food supply is faster to adapt and more resilient than ever before. Which brings us to the future. As our climate continues to change and new environmental pressures mount, agricultural science is going to be in a constant, relentless race against time. So I want to leave you with this provocative thought. As climate change accelerates, which crop will we need to genetically rescue next? Thank you so much for joining me in this explainer. Keep wondering, keep learning, and I'll catch you next time.